Thank you so much, Sabrina. Thank you so much to Arjun and Shalev for the kind invitation to speak. This is has been such an exciting couple of days, and I would love to be in Banff with all of you and actually have one-to-one interactions. So my lab is very interested in understanding how cells make decisions. I'm trying to change my slides. Just one second. Perfect. So our lab combines experimental and computational approaches to understand cell decision making. And in particular, we've been very interested in understanding two fundamental decisions: the decision to undergo cell division and the decision to undergo cellular differentiation. We are very interested to understand how these two decisions are ultimately very interdependent. Now, an important concept in cell decision-making is the idea that is very rare in biology, that a single gene or a single protein alone are responsible for cellular responses. So on the contrary, oftentimes it's really the ability for molecules to interact with one another and ultimately on how the concentrations and how the activities of these molecules are regulated in time and are regulated in space that are really key for cellular responses. I think cells do this best by coordinating regulatory networks in ways that allows them to discriminate between different signals and allows these networks to trigger different cellular states and ultimately different fates. So an extremely useful system to explore regulatory principles in decision-making are actually human embryonic stem cells, which have this magical propensity to differentiate into the three germ layers and virtually give rise to all the cells in the human adult body. These cells, of course, also self-renew, so they are also an awesome system to understand cell division. They are incredibly trackable and a fantastic system to understand how cells compute decisions and give rise to early fate choices during human development. Now, the model we use in the lab is mesendoderm differentiation of human embryonic stem cells, and this is driven by one molecule called BMP4. And upon BMP4 stimulation, these cells go from these tiny cells that grow in colonies to cells that are much, much bigger, about 10 times bigger, and are importantly able to migrate away from the colonies and live as single cells. Now, BMP is really a fantastic system to use here because not only we differentiate cells into mesoderm, so BMP is known to be a mesoderm factor, but actually about 3 days after BMP stimulation, we recapitulate really gastrulation. So we see populations of mesendoderm cells, of mesoderm and endoderm, and populations of extraembryonic layers. And so in a way, this is a very important system to mimic gastrulation in vitro. But what makes it extremely useful is that we can actually use mycopatterns and thereby confine these cells to fixed geometries. And when we do that, and this is, by the way, a technique developed at the Rockefeller Institute by Aria Wormflash, magic really happens. So what you see is the exact patterning that you see in a human embryo, where the outer layers are the extraembryonic layers in the embryo. The middle layers are really the mesendoderm. So we've got endoderm and mesoderm cells, and the inner center of these colonies are going to give rise to the ectoderm. So exactly as you would see in the human embryo. So we can now combine, of course, our mycopatterns and doing imaging in them with single-cell RNA-seq data and have this magic information of where each gene and each protein actually physically are. So we're having, a fantastic spatial and temporal resolution of these first decisions in human development. So, of course, to combine sequencing and imaging, we also need a lot of image analysis. And in the lab, we have been in recent years putting a lot of effort to use neural networks and track these cells. Mind you, these cells change dramatically in morphology. And so we are now able to track individual cells within these colonies as they have these dramatic changes in morphology. So let me tell you a little bit about what happens during BMP differentiation. So the changes in morphology were obvious. I showed you in the first movie, There are, of course, massive changes in gene expression programs where pluripotent genes are downregulated and lineage-specific genes are upregulated. There's global changes in epigenetics and the cell cycle remodels, whereby differentiated cells have much slower division cycles. Now, all of these are actually late events in differentiation, most happening about a day or two since each of these pluripotent cells sees BMP for the first time. So you basically need days for any visual changes in gene expression or in chromatin or in cell cycle regulation. So Alex, a PhD student in my lab, was very interested in understanding whether irreversible commitment to differentiation was also a late event, was also something that happened as cells progress through differentiation. And what Alex found was actually extremely surprising. And it was the fact that cells actually commit to differentiate within minutes. So the experiment that Alex did was very simple. She pulsed cells with BMP for just half an hour. And what you can appreciate hopefully here is that 3 days later, these cells have changed in morphology. They have upregulated these red genes here, so the mesendoderm-specific genes, and downregulated pluripotent genes, exactly the same way as cells that have seen BMP in a sustained manner for 3 days. Now, if the pulse of BMP is any shorter than this half an hour, absolutely nothing happens. The cells continue proliferating away, and all the pluripotent genes are upregulated. So she saw this, of course, for a variety of other genes, but importantly, when we use our micropattern system, you see that the pulse of BMP gives rise to exactly the same signaling, exactly the same first waves of gene expression, and exactly the same hallmarks of gastrulation that we see when we stimulate with sustained BMP. So this was extremely exciting. Now, how are these signals decoded by the cells? So the BMP is actually transduced by the so-called SMAD transduction network. SMADs are transcription factors. They have to go into the nucleus to transcribe genes. And if you don't have SMAD, even in the presence of BMP, these colonies just continue proliferating away and the cells don't differentiate. Now, we noticed that SMAD activation was sustained after BMP stimulation. But very interestingly, it had this switch-like behavior where the cells at the end, at the periphery of the colonies, got activated first, and then with time, more and more cells got activated. This switch-like behavior is also seen as we look at SMAD activity as a function of BMP concentration, and you can see that if you add a little bit of BMP, not much happens, but after some threshold concentration, you clearly see that more and more cells jump into this SMAD activated state. Now, switch-like responses and bistability are common in cellular transitions, and for the most part, they give rise to actually irreversible responses. And indeed, we see this. We see that if we activate SMAD pathway with BMP at maximum activity, even if you remove BMP to 0, these pathways remain on, showing a degree of irreversibility. Now, why irreversibility? Well, part of the answer is that these pathways are actually embedded in positive feedback regulation. So we found that one of the first genes that SMAD regulates is actually its own activator, the BMP receptor itself. So classical example where activator activates activator. But how are these signals actually decoded? So how is this upstream dynamics of signaling pathway actually translated into ultimately commitment to differentiation? So we run a RNA-seq experiment, and one of the genes with the highest score was actually not known to be a SMAD target gene, and this is this gene called GATA3. So we looked into it a little bit. So it was very interesting to see that GATA3 expression was very early, right? So at the protein level, after 2 hours of BMP, we start seeing cells expressing GATA3. But importantly, it had the same periphery versus inside of the colony, so the same spatial activation or expression as SMAD activation. And in fact, every time we see GATA3 at the mRNA level, we saw expression or activity of SMAD. The same dose of BMP that is important to activate SMAD is exactly the same that leads to expression of this gene. So these early genes are actually in not only GATA3, but in this little cluster of genes are actually mirroring completely what happens in upstream signaling. So that was very interesting. So we see that GATA3 is also embedded in positive feedback. So this is a chip experiment where we see beautiful peaks after BMP stimulation on GATA3 itself. And in fact, these nested feedbacks that we uncovered are predicted to help maintain a memory of this signaling and this expression of early genes. So as you activate the pathways with a little bit of BMP, even if you remove BMP to zero, these signaling pathways are going to be on, and the same with expression of GATA3. Now, positive feedback is obviously a common motif in cellular transitions. And so we were excited to see that potentially this is what gave rise to a switch-like irreversible transition and commitment to differentiation. So we were very excited about these dynamics, but we really wanted to understand whether GATA3 could actually be an early commitment gene that would tell us at the single cell level that cells had committed to undergoing differentiation, exiting pluripotency irreversibly days before we actually see any loss of pluripotency or any upregulation of lineage-specific markers. So this was very exciting for us to explore. What would you expect from an early commitment genes? Well, potentially, when induced, they should induce differentiation. This is exactly what we see. So we induce GATA3 in the absence of any differentiation cues, and we see upregulation of differentiation genes. And interestingly, we see this multi-lineage expression, very, very interesting for us. Now, conversely, if we knock out GATA3 with CRISPR, we see a delay in differentiation. So here's just a hand-picked handful of genes, cherry-picked to show mesendoderm differentiation, and you see that basically there is very much delay or a non-expression of genes in the knockout situation. So in addition, we expect an early commitment gene to regulate early targets. Now, this is a chip-seq over time of GATA3, and we see the common expected late targets of a fate marker like GATA3 used to be known for. So this means that, in early time points, you don't see GATA3 binding to any gene, and then you see these beautiful peaks after 48 hours. But actually what we see is that in about 3,000 genes, we actually see a very early regulation. So 6 hours GATA3 is sitting on these genes, and after which this regulation is lost. So GATA3 is definitely regulating genes very early during differentiation. And there is, the potential for network rewiring, which is extremely exciting. So we image, of course, a knock-in of GATA3, right? And what I wanted to show you, this is just I'm showing you the movie in a sec, but this is just a still image of a 9-hour. And you see that in this colony, every cell, every precursor cell is expressing GATA3, potentially at different levels, but I'll come to that. So initially there is no GATA3, we stimulate cells with BMP, and you start seeing, the cells spreading out, changing shape, and GATA3 late just really expressed especially in these flat cells outside of the main colony. So we actually see that the levels of GATA3 are predictive of fate. So this extra embryonic layer, so the cells, the flat cells in the outside of the colony are going to correlate very nicely with high GATA3 levels. The inside of the colony anti-correlate very nicely with GATA3 levels, so no GATA3. But very interestingly, when you have intermediate levels of GATA3, you actually specify mesendoderm. So it can be basically predictive of fate choice. So this is an example of just single cell tracing. These cells here have no GATA3. We specify, we have an intermediate level of GATA3, and we have a high level of GATA3. And what you can see is that the marker for mesendoderm, which is brachyury or TBXT, is really only expressed in these intermediate GATA levels. So one important thing we found out along the way was actually GATA3 is a pioneer factor that opens chromatin early in differentiation. So this is a complex slide, so I'm just going to try to lead you to this point here. So this is basically ataxic, what ATAC tells you is that the chromatin is opening, so at 0 hours and then 48 hours, 72 hours. And we combine this with CHIP and see that GATA3 is actually sitting at this chromatin later. Now, if you knock out GATA3, you see that this chromatin doesn't open at all. Okay. The reverse is also true. GATA3 is able to close chromatin. So this is ataxic again. So differentiation in this axis, you see that the chromatin is closing. GATA3 binds initially, but then doesn't bind anymore. And if GATA3 is not there, the chromatin doesn't close. It's very open. So we are super excited about this. This is basically just to show you in all the peaks. So these early peaks of GATA are basically sites where the chromatin tends to decrease, so tends to close. And this is perturbed when we don't have GATA3. Okay, so I'm basically trying to tell you that potentially we have found a new class of genes that are able to tell us way in advance that the cells have committed irreversibly to differentiate. And one of these genes is GATA3. So we think that it's behaving as an early commitment factor. If that's true, then this first experiment that my PhD student did, Alex, with a pulse of BMP, should basically be potentially compromised if we don't have GATA3. So this was the experiment. A pulse of BMP gives rise to differentiation. But when we don't have GATA3, this is absolutely compromised. So showing that GATA3 is very likely to be an early commitment gene. So I would like to finish by asking one last question, and it is, I'm basically showing you that cells irreversibly leave pluripotency and commit to differentiation. But how irreversible is actually this commitment? In other words, could we divert fate choice or not in this scenario? So the experiment that we did was very simple, and it was giving a pulse of BMP to the cells. And instead of putting the cells back in pluripotency media, we actually triggered another pathway. So we challenged the cells with retinoic acid for them to differentiate into ectoderm. And let's see what happens. So can the cells divert fate, or once they've committed to a fate? So cells that see BMP in a sustained manner become mesentoderm. Cells that see retinoic acid become trophoectoderm. A pulse of BMP, cells don't care, they will become mesentoderm. What happens now when we try to switch fates? And the cells are able to switch indeed. And this actually happens within a window of time of about 48 hours. So what I tried to show you today was that commitment to differentiation in human ES cells is an unexpectedly early event. We see that positive feedback in the networks that transduce these signals is really important to maintain a memory of the same signals. And we see that early genes, specifically a cluster of genes, really mirror the dynamics of that upstream signaling network. And we think that GATA3 might be an early commitment gene. Lastly, I showed you that while cells irreversibly leave pluripotency, they remain plastic to fate choice. And we are at the moment very interested in understanding what controls this plasticity with this plastic period. And just to give you an outlook to close, I think there has been a couple of models proposed for development, for fate choices during development. And the classical one has been very much influenced by morphogen gradients, where the position of a cell within the embryo really determines how much concentration of the precise signal they see, and therefore the pathway down in differentiation is very linear. Now, there is another model, and this has been very, very well shown for hematopoiesis, which is the model of cellular plasticity, whereby the first precursor cells are actually plastic cells. And with time, a fate is chosen and other two fates are repressed. And this is where we find basically the late commitment genes, or the fate genes that everybody knows. So we think that our data is potentially quite in line with this second model, whereby early on we have early genes that actually make the cells plastic to fate choice, and that with time, then one fate is going to be selected. And with that, I would like to finish by thanking, so all of the first work that I shown you was done by Alex, a PhD student in the lab. She's no longer with us, unfortunately. And the GATA3 early commitment as an early commitment gene is unpublished work done by Adrian, a fantastic postdoc in the lab. I would like to thank collaborators and the CRIC for being a fantastic environment. And thank you very much for your attention.
Thank you very much, Sylvia, for the wonderful talk. We have a question from Kaiser Loel. Great talk. Do you think that GATA is directly repressing the sites that are closing during differentiation, or do you think that it's some sort of indirect effect?
Thank you for the question. Potentially both. So we see very interestingly that one of the genes, or one of the set of genes that GATA regulates, are ectoderm genes, for example, PAX6. So it's basically promoting the mesendoderm branch and really repressing ectoderm genes, so not letting the cells become a fate any longer. And that at that site, we see beautifully that the chromatin is closing with time for ectoderm genes. So in that sense, is very direct. In other examples, we don't see as much as a direct cause. We are at the moment trying to, we have identified a set of candidate genes that we think might be binding partners, important for closing and opening chromatin. And we are basically knocking them out and seeing what happens to chromatin state at these sites.
Okay, thanks. Next question is from Dagan Segal, who asks, do you think you could model symmetry breaking with this system as well, for example, through asymmetric micropatterns?
We have explored different shapes of the micropatterns a little bit. I do think that probably the best way to go forward is using the new 3D system that was developed by Naomi Morris and Alfonso Martinez Arias, which is, of course, the gastroloid system. So human gastroloids are now available. I mean, we do them in the lab quite routinely now. We find that there are weaknesses in terms of really representing human development, but I think if you are interested in symmetry breaking, this would be the best system. And you've heard Prisca Liberali talk, and I think their lab is fantastic, has been doing fantastic work on this. So I would go for the 3D system in here.
Okay, thanks. There's a technical question here from Dee Hu. How many cells did you use for your chip, and how many sequencing reads did you use? The browser shot you showed looks very clean.
Yeah, thank you. So I don't know by heart, but we published cell protocols, star protocols. So please check us, because we noticed that a lot of people, even like very well-established labs of human development, were basically coming to us to ask for advice on how to do these chips. So we decided just to publish and make it available for everyone. So check it and please email me if you have any questions afterwards.
Okay, another question here from Gia Lee. Excellent talk. How are the other early progenitor markers such as the IL-1, CREB, PA changing by adding DMP4 or in the knockout of GATA3?
Yeah, so ISL1 is a direct target of GATA3. GATA3 is binding there at 6 hours. So you would expect this gene to come up after BMP stimulation, but in fact, you see a very big delay when GATA3 is not there. So it's actually upstream. So this gene is downstream of GATA3. So thanks for the question.
Okay, I'll just ask one last question of my own. Did you try modifying the size of these micro patterns? And are there any interesting changes in the spatial pattern when you impose different sizes?
Yeah, so I mean, there is really some type of a sizer, right? So if you have tiny micropatterns, you will form just the outer layer. So what we think are the extra embryonic layers. I mean, people now think that is basically amnion that we are forming. If you increase a little bit, then you start forming the outer and the immediately intermediate layer, and only a specific size will form the 3 layers. So the size makes a huge difference. The mechanism of this sizing, we actually don't know.
Okay, thanks very much, Sylvia. We'll move